Welcome to the afternoon session, everybody. The first speaker will be Colin from the University of Alberta in Edmonton, Canada. I met Colin through Peter, and I will tell you later how I met Peter. Colin is a great guy, very knowledgeable. He knows everything about the history of the models. It's not surprising because he has been a postdoc in Paul's lab. He had the privilege to test the danger model, and very interestingly, he has different conclusions from his mentor. And as you know, of course, to have a good discussion, it's important to disagree. If you agree about everything, there's nothing left to discuss. So I look very much forward to hear about Colin's version. And actually, he has done much more than testing the danger model. I think he has built his own model, which I don't know so much about. So. And I also realized that it's been 23 years since the last major model has been proposed by Polly. So maybe we'll get a new one today. In any case, you must welcome Colin.
Is that working? Yeah, okay. So thanks again for the invitation, Alex. It's a real privilege to talk amongst such great theoretical immunologists, and a bit daunting to discuss the danger model while Polly's sitting in the front row. Oh, you can come back to the front row. That's okay. This is where I'm from. In case anyone is interested in doing, for example, a postdoc, we live in the prairies here in a province that also has some nice mountains. So if anyone's interested, we are looking for a postdoc. I'll do some recruiting while I'm here. I'm going to take the opportunity to step up on my soapbox just for a minute or two here, because I think this workshop is in part a celebration of theory in immunology, and something we don't do that often. And I think there is a growing kind of values void in biomedical research. So what do we value in biomedical research? I think there are a couple of things that we would consider maybe universal values, and one would be description of new approaches to treat disease. We all value that, whether we do translational science or basic science. We all value description of new molecules, pathways, and cellular interactions. But I think some things that we don't tend to value as a community as much as we need to are linking diverse observations to come to new understanding of how a particular biological system works, and that really, in essence, is what theory is. And tied together with that, I think there's a decline in scholarship. We don't value correctly attributing concepts to their founders. And so why do the latter two matter? Why can't we just live with the first two? I think it becomes, as a culture, a failure to recognize that data is meaningless without interpretation, and theory is really the height of interpretation. Tied together with that, we end up suffering from Parkinson's law of triviality, which is basically the time spent on any item in an agenda will be inverse to the importance involved. And that really means we fail to understand that biology has a hierarchy of questions, a hierarchy of importance, from broadly important questions to minutiae of much more narrow relevance. So I'm going to step off my folks, and maybe because this is a workshop in theory, I'm preaching to the choir, I don't know. So just to give you a bit of background about myself and how luck and strategy is involved in why I'm here today, for example. So a long time ago when I had less gray hair, I started out in research with somebody named Nick Sinclair, who you probably have never heard of. So I will talk a little bit later about his contributions, but it relates to the idea or concept or theory of co-inhibitory signaling and tolerance. And after doing a PhD with him, and he's someone who's interested in broad theories. He was an experimental scientist, but also interested in broad theories. I decided to go to a purely experimental lab for a postdoc and found that not quite so fun. So I decided to strategize and try and find a lab that was interested in broad theories. And so I went to the top lab doing theories in the danger theory. And to get there, I wrote Polly a challenging letter, challenging her theory. And it was sort of a test to understand whether, how she approaches things. Was she interested in challenges to theories? And so it worked. And I spent a number of years in Polly's lab, and that was really a lot of fun. And I was fortunate that when I was there, Mel Cohn was doing a sabbatical, so I spent a lot of time at his office bugging him with a zillion questions. And more recently, I've had the good fortune of spending a lot of time discussing with Peter Bretscher, who's in the next door province in Canada, and we've done some collaborating together, so that's also been a lot of fun. So these are the people who've sort of influenced the way I think about science. And luck continued in that I've been able to participate in workshops with a group of immunologists who are also interested in broad theory and immunology. And I think we owe a really debt of gratitude to Zlatko for fostering theory in the Scandinavian Journal of Immunology and fostering discussion. And now your president here, Alex, I think he's continuing on the same vein. So thank you to both of them. So what I want to do is talk to you about what I call a development context model. And hopefully I don't disappoint you in that I'm not going to propose here a brand new theory. There's, this isn't like Peter Bretscher's T-cell help and Polly's danger model. This is trying to synthesize the novel ideas of the past and sort out what I think is useful. So it's an effort to synthesize what I think is correct in these minimal models and define the relative contributions of the mechanisms. So often theories are presented as being mutually exclusive, but I think if one looks at them, there's often something correct in each of them. And if we simply weed out the bad parts and put them together, maybe we have a more accurate picture of reality. So the problem that I'm going to talk about is what's traditionally called self-non-self discrimination and how the immune system sorts T and B cells into the ones that are maintained versus the ones that we want to get rid of, so typically the anti-self T cells. And this, of course, follows on from Burnett and Talmage's clonal selection theory. So a number of years ago in the Scandinavian Journal of Immunology, I tried to synthesize my viewpoint and called it the development context model. And so it built on the ideas that was proposed by Joshua Lederberg. So Joshua Lederberg was a Nobel Prize-winning microbiologist. He showed that bacteria could have sex, but he was much less known for his really great contributions to immunology. So he contributed many things to immunology, and probably the most important, I would say, theoretically, is the idea of central tolerance. So he proposed that as a lymphocyte develops in an organ such as the thymus or a B cell in the bone marrow, it's programmed to be selected out or made tolerant if it encounters its antigen at an immature stage of the lymphocyte. Not an immature stage of the animal, but of the lymphocyte. Each lymphocyte is programmed to die or be selected out if it encounters antigen before it's fully mature. And so I'll argue that I think that's one of the most important theories in immunology, if not the most important. Certainly there are aspects of the theory we can debate that didn't turn out to be correct, but in terms of tolerance, it's really one of the most important ideas. So in development context, tolerance comes from a co-inhibitory signal, which was proposed initially by Nick Sinclair, and that is that the antigen receptor, together with a co-inhibitory receptor, would lead to inactivation of lymphocytes. And the other aspect is immunity, so what turns on the immune response. And so this model is supportive of the ideas that PAMPs and DAMPs are promoting the immune response. They lead to co-stimulation, the opposite of co-inhibition, and leading to an APC activating a helper T cell to get the immune response going. So one thing you'll notice that's not on here, certainly in this 2006 version, is the concept that the immune response is started by T cell help. So, I'd spent a lot of time debating these ideas with Mel Cohn, and he wasn't able to convince me that T cell help was a critical determinant of self-non-self discrimination. I always thought it was the ideal mechanism for class control because it allows coherence of an immune response. To have it dependent on the interaction of two antigen-specific cells leads to a coherent class of immune response. So that's where I thought the mechanism played a role. But in talking with Peter Bretscher over many coffees and beers, and then reading his recent book, which I would encourage everyone to have a look at, he's made arguments to me that I think are fairly convincing that T-cell help is a critical aspect of self-non-self discrimination. So, what are the sort of postulates of development context? That the specificity of immune responses is controlled by somatic selection, so that's not controversial, but also by controlling collateral damage. So you can have the same repertoire, and the immune response can cause substantial collateral damage. So the specificity of the eventual effector output can be controlled to lead to a more specific immune response. So collateral damage is something that the immune response does because it's evolutionary beneficial, but if you didn't have control of it, you would have no specificity despite a somatic selection of your repertoire. So both processes are needed. Central tolerance is also part of this, and I would argue, the greatest contributor to the somatic selection that decides a self-non-self discrimination. In this model, the TCR and BCR signal alone is a weak activating signal. Inducible co-stimulation amplifies activation, so this is the positive context within this context model. And T-cell help controls class, memory, and detects the degree of foreignness to promote activation to very foreign entities. So the number of different helper cells you have to a particular molecule influences greatly whether you will get a productive response. And peripheral tolerance then is instead the result of inducible co-inhibitory signals. So that's a negative context, and it has some specific roles during development. And two, that I'm going to not talk about this any further unless people want to discuss it, that it does also demand that there would be some epitope-specific suppression by regulatory T cells. I think Zlatko has much more novel propositions about the purpose of regulatory T cells, and we'll probably hear about that later. And lastly, collateral damage must be controlled to achieve a successful self-non-self discrimination, and mechanisms here would include co-inhibitory signals and limiting the ability of antigens, for example, to be cross-presented on MHC class 1. So we don't want our B cells to be cross-presenting antigens from viruses that are not infecting them and leading to them becoming CTL targets and killing off those cells. So that's a form of collateral damage which would be evolutionarily not very beneficial. And similarly, we don't want our endothelial cells to be doing that. It would result in our destruction of our organs even if the endothelial cells weren't infected. So what are the differences from, say, the danger model? So in the danger model, central tolerance is not the major contributor to the somatic selection, as far as I understand it. The TCR and BCR signal alone is tolerance. It doesn't require co-inhibition. And there's no role for ontogeny-specific mechanisms of tolerance. That is, the tolerance is, the mechanisms of tolerance are the same throughout life. So how about, and one thing I should mention, when I talk about the danger model, I'm, just for simplicity, limiting the discussion to the danger model. There are related models, and Zlatko will talk about integrity, and there's someone named Jamie Cunliffe who has a morphostasis model, and these are related to the danger model, but for simplicity, I'm just going to talk about danger. So what about the model of Peter Bretscher and Mel Cohn? They're similar but distinct models, but they have some similarities. They're about the idea that, in essence, the immune system is a quorum sensing mechanism, that you need two antigen-specific cells to start an immune response. So a helper cell needs T cell help to become an effector T helper cell. So another T helper cell must also be seeing its antigen to get an immune response. And this idea proposes then that any antigen early in life will lead to tolerance through signal 1, and that's due to the absence of T cell help. So when T cells first come out of the thymus early in life, they're going to be at very low frequency or low density distributed throughout the body. When they encounter antigen, they're going to see it without a collaborating T cell to give T cell help, and they're going to become tolerant of that antigen. So how is that different from what I would suggest? So Peter will, this comment here is not specific to Peter's view, it's more specific to Mel Cohn's view of T cell help. And that is, in this T cell help model, central tolerance is not the major contributors to somatic selection. And again, I think Peter also would disagree with this. The TCR and BCR signal alone are tolerance, and that's again the same as in the danger model. And the key difference is that this T cell help model really proposes that the age of the whole organism, not the age of the lymphocyte as in Lederberg, that the age of the organism is a key factor or the central factor in generating tolerance. So any antigen that exists prior to immune system development should be treated as self. And Peter calls this the historical postulate. So if we compare the different models, which model is closer to reality? And I think graft rejection is perhaps an ideal way to test the different models. Why do I say that? I think one can study infectious disease immunology and perhaps generate some general rules, but there you're dealing with the competing evolution between the host and the microorganism. So there is a good chance you'll come up with microorganism-specific rules and not necessarily general rules of immunity. So I think graft rejection, where there is no evolutionary competition between the antigen and the recipient, might be the best type of system to try and look at what are the default rules of the immune system. And so the models we've talked about here have different explanations for graft rejection. So why are grafts rejected? In danger, it's because surgeons create damage. Surgeons are bad. They cut, they cause necrosis, the types of cell death that are going to trigger an immune response. In the T-cell help model, donor antigens were not present early in life to induce tolerance, and therefore you're going to reject a transplant from a donor that's where their antigens are different. And in development context, it would suggest that donor antigens were not present in central lymphoid organs to induce tolerance. That is, the Lederberg mechanism had not taken effect on donor antigens. And there is an important, but I would say lesser role for surgeons create damage. So if one looks in the literature recently, one would see that the idea of danger-associated molecular patterns is really quite prominent in the literature in terms of its role in rejection, and we've heard about some interesting studies earlier today. But I would argue that these reviews, certainly, of the topic kind of avoid the whole question of experiments that actually test the question. So they look at each individual danger molecule and try and inhibit it and see if you get better graft acceptance. But that type of experiment can't actually test the danger model. That is, it can't, if the inhibiting that particular danger molecule doesn't cause better graft acceptance, it doesn't disprove the danger model in any way, because it could be some other danger model or molecule. So quite a long time ago now, and Paulie's talked a bit about these experiments, I'm just going to discuss them a bit more. We tried to do experiments that would test sort of all three viewpoints. At that time it was two viewpoints, the T helper model and the danger model, as well as ignorance models, for example. And so we took these immunodeficient mice and gave them a transplant. And in essence, this is like doing a perfect immune reset. So in transplantation in a normal individual, I guess the closest thing would be someone who's getting immunoablated and then getting the transplant. So here we have a perfect immune reset because the recipient never had an adaptive immune system to begin with. So it's not an imperfect depletion of recipient lymphocytes. They have no lymphocytes. And we could put in a transplant, and shown here is an islet cell transplant as one example. And we could put that into the recipient and let it sit there and heal for many months, and then inject hematopoietic stem cells of the recipient type and ask if the transplant would induce tolerance. And by all theories, we really thought these transplants should induce tolerance. So in the danger model, because the grafts healed in, the danger signals have dissipated. And in the T-cell help model, because the antigen, the donor antigen, is present before the immune system develops. And so T cells would migrate out of the thymus. They'd have to become tolerant of peripheral self-antigens, and they should, in theory, then become tolerant of peripheral graft antigens. But instead, as theorists, we kind of got it wrong, and a miracle occurs, and the graft's rejected. So Some of them were rejected, some of them weren't rejected. And which ones weren't rejected? Well, it turned out to actually fit to pretty well to Lederberg's model. And that is the idea that it's not getting the antigen early in life that matters, it's early in the age of the lymphocyte. So if we had grafts where there were passenger cells coming from this graft that could go to the thymus, then the grafts would be accepted. But if the graft antigens just remained in the periphery, they were usually rejected. And so I'll give you an example here. Here's one type of experiment we did was to take a B6 nude mouse that's female and give it a single minor mismatched skin graft, so an HY male nude skin graft. So this is from basically a cage mate male, so all it has is the HY antigen. We let it heal in, and you can barely see that there's a graft there, the edge of this graft. And what we do then is put in a thymus transplant that's female. So you would have then, from the bone marrow, hematopoietic stem cells producing progenitors for T cells. They'll go to the thymus, develop into T cells. They go out into the periphery, and they have to become tolerant of self-antigens and presumably tolerant of the HY, single minor antigen in the graft. But instead, the outcome was rejection. And the only time we would see acceptance of these skin grafts was if the graft came from a hairy mouse. Why would hair matter? Anybody? Well, hair didn't actually matter. So it's just that when it came from a male hairy mouse, that meant the donor had a thymus. So there were passenger T cells in the skin graft that could migrate into the thymus and induce tolerance to the donor. So we started to think maybe skin is the wrong type of tissue to test the danger model. At least I was thinking skin is external to the body, and if we think about transplantation, the organs that are hardest to transplant and get acceptance are skin, intestine, and lung. All the tissues that are continuously exposed to the environment. They have microbes, they're at least with skin, they're exposed to scratching, for example. So maybe the danger model shouldn't be tested using that type of tissue. So we moved to using heart tissue or islet cell transplants. So they're transplanted internally under the kidney capsule. This is just showing you a neonatal heart transplanted under the kidney capsule. You can see heart tissue here beside the kidney. And those hearts beat, they function, and so we could put in a single minor mismatched heart transplant, heal it in before giving recipient female hematopoietic stem cells, and the outcome was that these healed-in grafts, single minor mismatched internal grafts, did in fact induce tolerance. So we're going, yay! We've shown that a graft can induce tolerance if it's healed in before the immune system development. The problem is even a fresh graft, that is a graft given after the immune system develops, that's mismatched by such a weak mismatch, does not get rejected. So it made it difficult to make conclusions about the role of danger signals in terms of triggering rejection, because the single minor mismatch isn't sufficient to lead to rejection of internal transplants such as heart and islets. So we could then ask the question, maybe danger is critical for rejection of a bigger mismatch, say multiple minor mismatches. So we switched to using a BAL-B donor, so it's MHC matched and multiple minor mismatched. And we give these BAL-B transplants, heal them into the RAG knockout recipient for many months, and then give recipient-type fetal liver cells. Or we can give the transplant after reconstitution with fetal liver cells. So these would be grafts with damage or damps, and these would be grafts without damps. And we could do the healing in for 9 months, 1 month, or another group where the graft's given after fetal liver constitution. And then we can look for priming and a CTL response induced by the graft itself. And what you can see is you see a nice CTL response to these BALB minor antigens if the graft was given after immune reconstitution, but you don't see a CTL response here when the graft was healed in before reconstitution. And so fresh grafts were rejected and healed in grafts were accepted, and they induced tolerance. So is the answer that it's danger? Well, it could be, but the problem is that, as Polly mentioned, these recipients become systemic chimeras and you have central tolerance, so it can't really test the question in terms of danger. And so this just shows you could see donor cells circulating in the recipient of the long-healed graft. So our next question would be then, do healed-in multiple minor grafts induce tolerance because of the lack of danger or because of chimerism and central tolerance? So for this, we used these multiple minor mismatched donors that were either wild type that could carry passenger lymphocytes that could migrate to the thymus, or we used RAG knockout donors and transplanted those. And again, looked at post-immune reconstitution, whether there was rejection or not. And as Polly alluded to, if you have a wild-type donor that can generate chimerism, and you can see the chimerism shown here, and these are actually, in this case, with heart transplants, it ended up being passenger chimerism with B cells rather than T cells, that those recipients that are systemic chimeras would tolerate their transplant. Whereas those without chimerism, these RAG knockout donors, about half of them rejected. So Polly argues that the half that didn't reject supports the danger viewpoint. And perhaps that's true. But the half that didn't reject, if you give them a second transplant, close to 3/4 of those remaining mice will reject the second transplant. So they hadn't, most of them, become tolerant of the healed-in transplant. So does this few mice that didn't reject here help support danger? I don't think so, because if you think about the length of time that these mice have been going, so the many months of healing in the transplant, then many months post-immune reconstitution, I wouldn't be comfortable about concluding about the role of danger here unless I did the control of a third-party transplant. So are they sufficiently immunocompetent for all of them at this age to still reject the multiple minor transplant? That's why I'm not yet comfortable concluding that this is supporting the role of danger. But it does clearly support the idea of Joshua Lederberg and central tolerance being critical to deciding what we reject and what we don't reject.
How long time do you have between the first and the second transplant?
Well, the first transplant is given many months before fetal liver reconstitution, and then the second transplant is given more than 100 days after immune reconstitution. So it's probably 6 months on average between the two, or more, actually more. So probably 10 months. A second heart transplant or a second islet in this case, islet transplant.
So between the two islet transplants, it was like 6 months?
More, yes, yeah. So I think contrary to predictions of both the danger and T helper models, healed-in pre-immunocompetence grafts induce immunity rather than tolerance. And so one might argue that healed-in grafts continue to send danger signals that prevent tolerance and trigger immunity. When we looked at this in healed-in skin grafts, we couldn't find any elevation in inflammatory cytokines in a healed-in skin graft. So what about when we did these internal transplants, so heart transplants and islet transplants? So can the length of healing time determine immunity versus tolerance, and are healed-in grafts sufficiently normal to induce tolerance? So we took these rag knockout recipients, we gave them a male heart in the kidney. We then reconstituted 4 days, 10 days, or 6 months later with female fetal liver cells. And then a few months later, we could do a hemisplenectomy and ask, did the transplant, whether it was healed in long or short, induce a priming for a CTL response against the donor? And if there wasn't a response, could we immunize and test whether they're tolerant or simply ignorant of the transplant by doing then a second CTL response? So if you look at the first CTL, that is asking whether the graft itself primed the immune system to the donor, you can see that no matter how long we healed the graft in, the graft itself didn't trigger much in the way of a CTL response against the donor. But you do have to remember, even 4 days of healing in this case is actually a fair length of healing, because when you put in fetal liver, it takes about 3 weeks before you start seeing any T cells come out of the thymus. So this graft really is healed in for 3 weeks plus 4 days. So it's had a fair bit of healing. If you then look at immunizing these recipients and looking at a CTL response, can you immunize against the donor antigen, or have they become tolerant? What you do see is the longer healed transplants tend to give you tolerance, whereas the shorter healed in transplants, you could get some priming in these recipients. So I think that argues that DAMPs do reduce your ability to get tolerance, and there are other data in the literature that would be supportive of that viewpoint. So I think rejection of healed-in, danger-free multiple minor grafts and tolerance of single minor grafts is what we see in this data. And does this indicate that danger signals trigger rejection of a single minor graft? As I mentioned, the answer is no. Even a fresh single minor internal graft with danger signals doesn't get rejected. And this just gives you an example. In this study, you could take a female recipient and pre-sensitize it against the male minor histocompatibility antigen, and then give it an HY mismatched islet cell transplant, and those transplants don't get rejected, even in a pre-sensitized recipient to this single minor antigen. So the question then is, why are dangerous single minor grafts not rejected? Well, healthy multiple minor grafts are rejected, which seems to be a paradox. And I think the best answer there is that the number of foreign antigens, which would equate to the number of different helper cells one would have against those antigens, is a more critical determinant of immunity versus tolerance. So danger predicts rejection also of self-peripheral tissues generated by, caused by newly generated T cells. So in the danger model, it doesn't really matter whether the antigen's foreign or self. It's about the context of presentation. It does matter in the sense that self-antigens later in life, you're going to have been inducing tolerance to them all the way along. But if you could have an immune system develop in the presence of a self-antigen that's presented in the context of danger, it should be rejected based on the rules of danger. And there have been old studies in the literature kind of looking at this question. And so the first one was done by Triplett, where he did removal of the pituitary in frogs and grafted them. and showed you could get a rejection or infiltration of syngeneic grafts. And so this sort of argued that peripheral tolerance, and that tissues induce tolerance to themselves. So this is the concept that if you remove a tissue, a peripheral organ, you should be removing peripheral tolerance. And if you put that tissue back in later in life, it should be treated as foreign, and you should reject it if there was peripheral tolerance. So we did do some experiments where we took a female mouse and took out her ovaries and then waited for 35 days so newly generated T cells could come out of the thymus. They would not be able to encounter ovary antigens, and therefore if there was peripheral tolerance to the transplant was absolutely critical, the ovary should be rejected. So here you can see that when we transplant ovaries under the kidney capsule of a female mouse that had lost her ovaries over a month ago, that ovary transplant doesn't get rejected, even though it's into an immunocompetent recipient. And this is into a non-overectomized recipient. And yet, in fact, you can transplant ovaries into a B6 male mouse. So the B6 male never had ovaries. And the immune system doesn't reject those ovaries in the recipient. Whereas if the donor tissue is mismatched, that is, it contains foreign antigens, those ovaries are rejected. And we even, at one point, used nude mice, female nude mice, overectomized those, and put the ovary in as a transplant, and then reconstituted those mice, and again, they didn't reject the ovaries. So together, so these data, I think, argue that, yes, there can be peripheral tolerance, but that it's a much more minor component of self-tolerance than central tolerance. So there's literature on similar types of experiments where you can see some infiltration of a syngeneic tissue, but it's not something that's going to occur in all experiments. So it's quantitatively, the contribution of peripheral tolerance is much less. So why are grafts rejected? I think the danger viewpoint is not a general solution for understanding transplant rejection. Healed grafts without DAMPs are rejected when MHC or multiple minor mismatched, and it can't explain why a fresh single minor graft is not rejected by immunocompetent recipients. But the presence of danger-associated molecular patterns, I think, blocks tolerance, and that, in fact, perhaps danger molecules are about preventing tolerance more than they are about turning on a response. So if we look at just sort of an overall model, you have central tolerance, which is really filter one of the immune system, sorting out, doing the somatic selection, sorting out self-reactivity. In between here, you have a population of T cells that we would call recent thymic immigrants that haven't yet undergone the second filter of tolerance, which is peripheral tolerance, and that the main deciding factors that these cells must go through are first a somatic selection in the thymus, and then in the periphery, peripheral tolerance would be dictated by the number of foreign antigens within that molecular complex, which determines how many helper T cells are responding, and then DAMPs and PAMPs would then help prevent tolerance to those antigens. So the T helper model postulates a window of tolerance early in the life of an organism, and I think these experiments where the graft was present before the immune system development rules that out as the major criterion for the decision between immunity and tolerance. Because immunity can occur to antigens which were prior to immune system development. So that raises the question, does the fetal neonatal period contribute any unique properties to promote tolerance? And I think it does. So I think the Bretscher and Colin type of model, where early in life there's this tolerance window, I don't think it's an absolute, but that, of course, as T cells first come out of the thymus early in life, T cell help will be reduced compared to in the adult, and that will help promote tolerance. But it's not an absolute requirement to achieve tolerance. And I'm just going to give you another example where I think the age of the animal really does promote tolerance, and I'll explain why I think that does so. So we did an experiment where we took hematopoietic cells that were PD-1 deficient in the co-inhibitory receptor PD-1, and injected those into RAG-negative recipients that are otherwise syngeneic, except for they have PD-1. And that resulted in a severe and drastic autoimmunity. So it was systemic, multi-organ inflammation. However, if you do that same experiment into a neonate, you don't get autoimmunity. So that's shown here. So in an adult RAG-negative recipient, as soon as T cells start coming out of the thymus, which is about here, the mice rapidly get very ill. But if you do that into a neonatal animal, almost all of them are protected from disease. And so what is it about the neonate that protected these mice from autoimmunity from syngeneic cells? I don't have definitive evidence, but the evidence we do have would suggest it's about the lymphoid organs in which T cells are maturing. I'll skip that. So in a neonate, the organs that the lymphoid tissue that T cells are migrating out into are immature themselves, and so that helps promote tolerance. So in the development context, signal 1 is a weakly activating signal, and this signal 1 can become much more activating when competition is low, such as in lymphopenia. And so lymphopenia is known to be associated with autoimmunity, but it's also something that naturally occurs during the neonatal period or the fetal period in humans. And so we hypothesized that PD-1 as a co-inhibitory molecule promotes the establishment of tolerance by preventing the normally low level of signal 1 that tonic signals of peptide MHC generate. So these aren't agonist antigens, but they're perhaps the same peptide MHCs that cause positive selection, that these signals in the setting of lymphopenia are controlled by the co-inhibitory signals to keep them at a threshold that doesn't lead to full activation. And so if we have lymphopenia and a lack of a co-inhibitory receptor, you now move that threshold to where a much larger fraction of cells can be activated. So how could we test this? So we used a model system using the Maryland mouse that was generated in Polly's lab with Olivier Lantz. And so here we can have a system where we have a defined monoclonal T cell. This T cell recognizes the HY antigen in IAB, and we could have it where it's wild type or lacking PD-1, and mix thymocytes from these two different mice and put them into a RAG knockout female mouse. So the important thing here is there is no agonist antigen. The recipient's female, and this TCR only recognizes the male antigen HY. And we could put it into the female mouse or a female mouse that lacks MHC class 2, so it's a C2TA knockout, to ask about whether recognition of self-peptide MHC was important. And what you can see here is that in the absence of agonist antigen, but these are lymphopenic recipients, the T cells, looking at violet dye here, T cells proliferate in these recipients. And the PD-1 deficient cells proliferate more and come to predominate in the recipient compared to the wild-type cells. And this requires recognition of self-peptide MHC. So if you transfer the cells into a mouse lacking class II, they don't divide. So I think this argues that the PD-1 co-inhibitor receptor is needed to control lymphopenia-induced proliferation to our own tonic peptide self-MHC signals. And this is particularly important in setting up tolerance when T cells first come out of the thymus into a lymphopenic neonatal or fetal mouse. So as a sort of simplified model here, we can think of these two filters again, the thymus and the periphery, where T cells coming out of the thymus into an adult recipient, they go out into these large empty lymph nodes and they homeostatically proliferate. You get lymphopenia-induced proliferation. And to establish tolerance in this setting absolutely requires PD-1. During the neonatal period, in contrast, the T cells are going into these much smaller lymph nodes, and they don't have the tonic signals to sufficiently proliferate and cause autoimmune disease. So the neonatal period helps constrain lymphopenia-induced proliferation. So this would predict then in, say, a normal PD-1 knockout mouse that they would have upregulated in a normal mouse, not a PD-1 knockout, they would upregulate PD-1 in proliferating cells during the neonatal period. And that's actually what we see when we use a mouse where we can track newly generated T cells that express GFP due to the RAG2 promoter. So only newly generated T cells express GFP and they lose it as they age. What you can see in these neonatal mice is that PD-1 is upregulated on cells that have lost their GFP, and that those cells are dividing. They're Ki67 high and they're CD44 high. So in a neonatal mouse, PD-1 is upregulated on cells that are undergoing lymphopenia-induced proliferation. So it raised the question for us as an adult, are there these newly generated cells that could cause autoimmunity, and are they immediately controlled as they come out of the thymus, or are they maintained as autoreactive cells as they are exported into the thymus? So what we did was sort out newly generated GFP-positive cells out of the spleen that lack PD-1 versus established T cells and transferred them into RAG-negative recipients. And just as one measure of disease, you can see here that recent thymic immigrants would cause weight loss and disease in recipients, similar to injecting thymocytes, whereas non-recent thymic immigrants or established T cells, they didn't cause nearly as much disease. So I think the importance here is that we have two filters of tolerance, central and peripheral. And if you take cells that are stuck in between the filters, they've gone through central tolerance but not peripheral tolerance, you can get increased autoimmunity with those cells because they haven't undergone peripheral tolerance. So establishing tolerance with newly generated cells requires a peripheral tolerance mechanism which is involved in setting the thresholds for activation of those T cells. So the function of signal 1 in co-inhibition, again, signal 1 I think is typically a weak activating signal, and that other signals determine the outcome. Tonic signals become activating when competition is lacking in lymphopenia, for example, and that co-inhibition is needed to establish tolerance under conditions of low competition. And lastly, that co-inhibition also increases the specificity of responses by limiting collateral damage. That is, the ability to recognize these tonic signals in an activating fashion that would cause collateral damage. So I've talked about co-inhibition, and you've all heard about it because of cancer immunotherapy and its great success in treating cancer. But where did this idea come from? So if we look at major concepts in self-non-self discrimination, this is my selective timeline of sort of important major concepts in self-non-self discrimination. I think one of the most important uncelebrated ones is that of Joshua Lederberg in central tolerance. And then Bretscher and Cohn's model of T-cell help in 1970. Lafferty and Cunningham in 1975 proposed the idea of co-stimulation. Janeway with pathogen-associated molecular patterns. And of course, Pauli with damps and co-stimulation in danger. But you don't see co-inhibition anywhere on this list. So it was in 1971 where Sinclair and Chan proposed the idea that the immune system is not made tolerant by signal 1, but by co-inhibitory signals. And so this is the figure from their paper. And what you can see here is the idea that the antigen receptor here would be an activating signal, and that the inactivating signal came from a putative co-inhibitory receptor. And in this case, back in 1971, they were focused on B cells. And so this paper proposed that a B cell would have an inhibitory FC receptor that would inhibit activation. And we now know, of course, that's true, FC gamma R2B1. And I think amazingly in this paper, they predicted that blocking the co-inhibitory signal would be useful in tumor immunotherapy. And that has proved to be the case. And being of theoretical nature, Nick Sinclair wanted to think of whether this mechanism might be something that's broadly applicable, or is this something just unique to a B cell. So he thought it must be a general mechanism that the immune system is shut off in a feedback mechanism through co-inhibitory receptors. So he proposed it would also work for T cells. In papers in 1990 to 93, and a lot of these studies are conceptual papers were published in the Scandinavian Journal. So the Scandinavian Journal has been fostering theory for quite a long time. And so he coined this term co-inhibition and postulated that peripheral tolerance was the balance between co-stimulation and co-inhibition. So I'll end there and just want to thank a number of people who did these experiments, some of which I showed you. Some of the old experiments were involved a number of individuals shown here, and Chris Elistad worked on tonic signaling, and I'll just thank all the agencies for funding in these difficult days to get funding. So happy to answer any questions.
Thanks a lot, Colin, for the talk, not for respecting your time. I guess it was a way to escape questions. No. From Colin. Okay, so I think I want a lot of discussion, but we have a talk by Peter soon. So let's say 2 or 3 questions, maximum 10 minutes, and then we keep more continued discussion later. Question to Colin. That was complicated. Polly. Please make the question simple.
Yuck. I'm going to start with saying I think it's a little bit unfair to say that the danger model doesn't stand on the shoulders of Lederberg. So the original birth announcement of the danger model was a 50-page paper called Tolerance, Danger, and the Extended Family. And of those 50 pages, probably 30 of them were about central tolerance. So the danger model very much stands on Lederberg. You start with central tolerance. Now, personally, I don't think that central tolerance is there to induce tolerance to every antigen in the body. I think its function is to induce tolerance to what dendritic cells present, because once you tolerize to dendritic cells, then you respond to whatever else they present, right? The problem is, as we showed in a Nature paper in 1986, that if you allow activated dendritic cells to go into the thymus, they still induce tolerance, right? So what happens there is it's because, as Lederberg said, as we wrote in Nature, there's a stage in T-cell development where if they see antigen, they get tolerized, which means that everything expressed in the thymus will induce tolerance. Danger model never said that that wasn't true. So it's a little bit of an unfair reading.
I didn't say the danger model said that's untrue. I said that the danger model says it's quantitatively less important.
No, it never said that. It never actually said that. So as I said to Peter, don't put words in my mouth. And if you actually can back that up, it would be really great to see.
Okay, but you clearly agree on central tolerance. What about peripheral tolerance? That's where the disagreement starts.
So I think we don't disagree that much. I think Colin says that DAMPs would inhibit or make it difficult to get peripheral tolerance, yes?
Yes.
So we both agree on that one. I think where we may disagree, but I actually think we don't disagree there either, is in the number of foreign antigen idea. So, people often ask me, why don't grafts between identical twins get rejected? Or why don't you just reject your skin when you get a cut? Mm-hmm. And of course, the point there is that you need both alarm signals to activate APCs, and you need a sufficient number of cells, T cells, to recognize that tissue to be able to reject it before it heals in. So I think if we put those together, we would actually have a model that was very similar. Now, where I think you're right that I haven't given enough importance to is in the original idea that signal 1 without signal 2, agreeing with Malcone, that signal 1 without signal 2 was a tolerogenic signal, and that you needed to overcome it with co-stimulation. But I've come around. I am now just about as much of a Sinclair advocate as you are. I don't care, really, if tolerance is induced by signal 1 without 2, or by signal 1 with low 2 and high co-inhibition. What I care about is that tolerance is induced by cells that are healthy and not by cells that are sick. And I can imagine that cells that are healthy find it in their advantage to induce tolerance and therefore have lots of co-inhibitory signals. And that cells that are sick find it to their advantage to turn on an immune response and would therefore downregulate their co-inhibitory signals. So maybe we should write a theory together.
Okay, thank you.
Okay, let's stop here the discussion for the time being, Colin, if you'll allow just to, and then again, there will be more time later. So thanks again to Colin. And the next speaker will be Peter Bretscher. I'm very grateful to Polly, not only for making the tangent model, but also for making a historical perspective on the models, which allow me, like, when was it, 5 years ago, 4 or 5 years ago, I was in a conference in Greece, and I sat on the bus going to the restaurant with a guy I did not know from before, and we just said hello to each other, and then he said, Peter Bretscher. I said Bretscher. These names tells me something. And that was because of you, Polly, because of the two-signal model by Bretscher and Colin. And Peter was kind enough to mention to me that he was the guy behind the model. So then I think we spent the rest of the conference discussing theories and much after that as well. Yes, so Peter is the guy behind the two-signal model, and I guess you will talk about that, don't you? Lymphocyte operation. Yes.
Sorry, can you hear me up at the back? Thank you.
So it's a privilege to have you here, Peter, and I'll let you talk.
Well, first of all, thank you for the invitation. I have to tell you, I'm going to make it slightly personal, but Colin already gave a little bit of background about his personal story. I live in Canada, and the nearest city is Edmonton. It's 556 kilometers away. And there's not very many people to talk to in Saskatchewan. And so about 4 years ago, I actually took a bit of my salary and decided I had administrative leave, so I decided I'd go around and try and invite myself to places without any obligation on the people on the other side or on me, because I was paying all the expenses. So I asked Colin whether I could come along and chat some immunology with him. And I went to one or two conferences, and on more than one conference that I went to, there was this gentleman called Alex Corfay. So I realized he went to lots of conferences. Anyway, I felt that being in Saskatchewan, I wasn't really managing to get immunologists to sort of consider what I was saying. Not to accept it, but to consider. So these two very nice people, I said, well, what should I do? And they gave me exactly the same advice, though one was a Canadian and one was Swiss-Norwegian. And they said, write articles for the Scandinavian Journal of Immunology and send it to that editor, one of the editors, Zlatko Dembik. And I did, and I'm not a great reader of the literature, unfortunately. So I didn't really realize that there were these discussion forums. And when I discussed this with Zlatko, he explained to me that Polly was partly the instigator of these discussion forums. So I think that we are really suffering from a lack of discussing big ideas in immunology. And we would do much better if we collectively got together. And I think this is a unique type of workshop. And I'm very, very grateful to have been invited. I'm going to use PowerPoint, although usually I've got a slight predilection to using the blackboard in chalk. But the reason for using PowerPoint this time is because I'm aware that one should only make about 3 or 4 points in a talk of 3/4 of an hour. Yet I really want to give a big sweep on the concepts in immunology. And so I have been to some, I'm not going to say mine is very good, but I have been to some wonderful lectures where everything is quite clear during the lecture, and then I get out and I can't remember the clarity anymore. The pieces aren't together. So I actually thought that if I did PowerPoint, I could put information there. If anyone wanted afterwards to try and recapitulate what I said, they could go back to the PowerPoint. So I hope that people can have access to the PowerPoint. Lastly, I want to talk a little bit about different types of theory making. I tend to like trying to face a problem and trying to find whether there are one or two assumptions or postulates which are clear, which can be related to the problem I'm addressing or trying to address. And whenever I've made a theory, if it's not in the title, it's certainly in my mind that it's a minimal theory. I'm not saying what actually occurs. I'm saying These principles allow a lot of things to make sense and be understood, but it's always a minimum theory. And so when people come along and say, but it doesn't account for this, I'll say, well, it wasn't designed to. But it gives a framework. So I just want to make 2 points about remarkable cases of where there have been really remarkable insight. One was the structure of DNA, which is really rather simple when you look at it. But if anyone goes to a lecture on DNA replication and mismatch repair or whatever, it's terribly complicated, but still it's all done within this very simple concept that was put forward. And the other obvious example is immunology is often called a field of complexity, and yet all our complexity is consistent with clonal selection theory, which was made by people who thought that some simple ideas could contain a useful step, although it's not, of course, describing the whole immune system. So this is a pointer, is it? So what do I have to do? This red one. Okay. That, and then advance it. Yeah. Okay, so that is the subject. And so this is really related to what I was saying before. I want to be very clear what my perspective is. I'm trying to say and explore the idea that signals from stress and PAMPs tissues can modulate immune responses. There's just no doubt about it. It would be someone very unwise and ignorant who denied it, and it makes a lot of physiological sense to me. So the critical question really in this session about what initiates immune responses is danger, including PAMPs, required to activate CD4 cells and so initiate immune responses, or is lymphocyte cooperation? So I'd like to give a little bit of historical perspective, and Colin talked a lot about this. So Lederberg, in his incredible paper in 1959, suggested that stem cells, what am I doing? Stem cells give rise to cells which are paralyzable on interaction. There's an internal clock, and if after a few days they haven't interacted with antigen, they become inducible. I don't know. And Lederberg suggested, sorry. It's all right. So Lederberg suggested that Burnett and Fenner's original idea could be extended, and that the presence of self-antigens early on in the lifetime. Whenever I push that, it moves it. I'm probably... Yeah, sure. All right, so... sorry. Okay, all right. Sure. Thank you. So this historical postulate, which is that the idea is that the self-antigens are present early on in development, usually before birth, and are continuously present thereafter. And in this way, in a manner that I think people understand, he accounted for central tolerance. Now, I do agree that central tolerance is about 95 to 97% of the solution to tolerance. But there are some antigens which aren't sufficiently expressed in the thymus to cause central tolerance. We need a mechanism of peripheral tolerance. There are, of course, peripheral antigens in the thymus, and they affect tolerance, and partly by deletion and probably by inducing T regulatory cells. I'm not going to say much about air except to make one point, which is that which I'll come back to in a moment. And what was not recognized by Lederberg, or not postulated by Lederberg, was that there was competition between the processes leading to immunity and unresponsiveness at the level of a mature lymphocyte. So in 1907, Melkon and I came up with this model that for mature lymphocytes are generated after self-antigens appear, essentially, and they're generated, those with the receptors anti-S are generated, the first one that's generated can only interact with one antigen alone, and a single lymphocyte, according to this view, is inactivated. So anti-S cells are wiped out as they're generated one or a few at a time, whereas you require collaboration to get an immune response. So a foreign antigen is different from a self-antigen in not being present early on in development. And as in the absence of the foreign antigen, lymphocytes with receptors specific for the foreign antigen can accumulate, and then when the antigen impinges upon the immune system, the antigen mediates cooperation. So this was what we proposed in 1970, and we had to explain a little bit of in terms of signals, which weren't really discussed much at that time. We suggested that antigen binding to the antigen-specific receptor would generate signal 1. Somehow, a target precursor cell which was being activated had to know that there was a helper cell because you required cell cooperation. So this model obviously was made at a time before anything was known about the T cell receptor. So the helper or collaborating cell had to somehow send a signal on recognizing antigen to the target precursor cell, and we suggested that this might be mediated by short-range molecules or by a membrane-membrane interaction. So that was sort of envisaged that interleukins and costimulatory systems might exist. So that was our model. And then once it became clear that T cells don't see native antigen but peptide MHC complexes, Lancevica came along in 1985 with a lot of evidence and a very beautiful model explaining how you could get BT cell interaction, which will involve the linked recognition of antigen, despite the fact that the T cell sees a peptide of the carrier. So that stage of very great paradox was an anti-hapten B cell could be only activated by a T cell specific for the nominal carrier if the hapten was attached to the carrier, and yet it was clear that the T cell, in order to recognize the antigen had to be cut up into little bits, and therefore the hapten would be dissociated from the peptide. So Lancevica's idea was that the anti-hapten B cell only takes in the hapten carrier conjugate, processes and presents it, and so the T helper cell will only help a carrier when it's specific for a nominal antigen to which the hapten is conjugated. So predictions of this one lymphocyte, multiple lymphocyte model for the antigen-dependent inactivation and activation of lymphocytes was that all lymphocyte activation required lymphocyte cooperation. And we proposed, there wasn't really evidence for it, but we proposed that the activation of CD4 cells themselves required CD4 cooperation. So in time, quite some time, people started finding that B cells, of course, require T helper cells, and the absence of T helper cells, the antigen can activate the B cells. So Norman Kleiman's work was very important, and Chris Goodnose. And for CD8 cells, similarly, it was found out that the activation of CD8 cells, at least some, requires T helper cells. And in the absence of T helper cells, antigen can activate these CD8 cells. That was Keenan, Foreman, and Goethe and Maxinger. So what all these observations led to was a recognition of the critical importance of what controls whether CD4 cells, naive CD4 cells, are activated or inactivated. And we had suggested that CD4 cells to be activated by antigen requires CD4 collaboration, and we did a lot of experiments to try and make that case plausible. So there are a whole lot. Some other people did too, the Goloni et al., but actually this has been an area where people haven't really done many experiments. But you can see there are quite a few papers there. I'm not going to go into what they showed, except to make out one point which I think is conceptually really critical. And that is that if you have a target CD4 cell, which you're going to activate, which is specific for the nominal antigen R, it can be helped by T cells specific for the nominal antigen Q, where Q and R are chosen not to cross-react, only in the presence of Q coupled to R as a conjugate. And that's very important because we thought that if that was not the case, it means that a T helper cell specific for foreign antigen, F, could help in the activation of a CD4 cell specific for peripheral self-antigen, PS, and therefore undermine the inactivation of the peripheral CD4 cell. So I hope that people do feel free to wave the hand if I'm not clear on something. So Janeway came along in 1989 with his proposal, and he did it in the context that CD4 cells, whether they're activated or not, determines whether B cells are activated or not. And he's made a generalization that when you take purified proteins, it's very difficult to actually make an immune response to them, and that you usually put them in complete Florence adjuvant, which you call the immunologist's dirty little secret. And he suggested that the activation of T cells requires two signals, signal 1 and a second co-stimulatory signal, which will only be expressed if a PAMP binds to a pattern recognition receptor. And that's what I've been saying. And then one of the weaknesses of Janeway's proposal, as far as I was concerned, and I presume, and I'm sure as Polly thought, was that there are situations where PAMPs aren't obviously around, and you can get rejection. One obvious example are transplantations. So she explained why transplants from a front vertebrate can be rejected, but she also brought on a number of other things. She explains how adolescent antigens can induce tolerance during adolescence. However, I would say that from what I understand of her model, you'd expect all vertebrate antigens administered under non-dangerous circumstances to inactivate CD4 cells. And one example I gave, which we've already touched upon, is sheep erythrocytes, and we already had a bit of a discussion about that, where we can come back to. So the PAMP in danger models are represented here. You need danger or PAMP to get the critical co-stimulatory signal that's required to activate the naive CD4 cell. So I came up with a model in 1999. This model, I should say, was consistent with everything we said in 1970. It was trying to be, it didn't deny the 1970 model. It tried to make a more explicit model of what was going on, consistent with a lot of evidence that had accumulated in the, almost 30 years, and that still had the feature that I liked of the 1970 model. And it was this model. So it was a two-step, two-signal model. And what I envisaged was that in step 1, an APC, which could be a dendritic cell or macrophage, would cause, through co-stimulation and signal 1 and signal 2, the T cell to divide, and that after division, The CD4 cells that have been stimulated to divide would crash and die, to be dramatic and clear, unless they went through step 2, which required the step 1 prime cell to interact with an APC, which is a B cell in this case, which had been activated to express the requisite co-stimulatory molecule to get this co-stimulation of the second step. So in this model, you've got actually 3 cells, antigen-specific cells, interacting in step 2. So it still explained peripheral tolerance, a single cell interacting with antigen in step 1, a single antigen-specific lymphocyte would interact with an APC and it would multiply and die. So a single cell would be antigen-specific lymphocyte would die, or CD4 cell. Whereas for activation, you required lymphocyte cooperation as expressed in step 2, and so it could still explain peripheral tolerance. So what I could regard as the positive features of the model is that it accounted for T-cell cooperation, a lot of observations on T-cell cooperation. This being a B cell in step 2, the APC in step 2, means that actually if that's a B cell, then the B cell will take in antigen specifically and can present 2 peptides derived from the same antigen to mediate the cooperation. If the APC has to be an antigen-specific B cell, it means that an immune response to a foreign antigen for which the CD4 affected T cells cannot activate a B cell specific for another antigen, and so you require linked recognition if it's a specific B cell. Whereas if it was just any old APC which could take in 2 antigens, that APC could take in a foreign antigen to which there was a response being made and a peripheral self-antigen, and so you'd undermine peripheral tolerance. And that is undesirable. So I say that if you've got an antigen-specific B cell in the second step, you've essentially got a means of ensuring you don't get interference. So you've got non-interference of self-non-self-discrimination by responses to foreign antigens on the inactivation of CD4 cells specific for a peripheral self-antigen. Yep.
What do you do about a cross-reactive antigen?
I'll come back to that if I might. Yeah. Sure. So there's one problem in this model, the way it's been proposed, which and that is that you actually require, according to this model, the way it's written, a CD4 helper cell to activate the B cell, to activate the step 1 prime CD4 cell. And yet, where does that CD4 effect helper cell come from? Well, according to this model, it requires an activated B cell to be activated. So you've got a priming problem. Where does the first activated cell come from, whether it's a B cell or a CD4 helper cell? And this is like the problem of you need life to beget life. You need a ribosome to make a ribosome to make ribosomal proteins. Where does the first ribosome come from? So I think there are solutions to this. I don't think that a solution is preeminent, but I'll just give one example because I think that if we don't have any potential solution, we're in a problem. So I would say that the CD effector cell there can be replaced, and although it'll be much less efficient, by a step 1 prime cell to get the ball rolling, and then you can generate an immune response. So so I would like to explain a little bit that why I'm uncomfortable about PAMP and danger theories. It's lovely that the activation B cells and CD8 cells is controlled exquisitely by CD4 cells. and that's consistent with our original two-signal model. If CD4 T-cell activation is not so exquisitely controlled and violating the principle of non-interference, I feel we're building cathedrals on sand. So if self-non-self-discrimination isn't precise at the level of CD4 T cells, it'll reflect on the activation of B cells and CD8 cells. And I couldn't help quoting Einstein here, everything should be made as simple as possible, but not simpler. Forgive me. I also would argue that there are many vertebrate antigens that are not expected to have PAMPs, and which, if given under non-dangerous conditions, are immunogenic, and I feel that's a problem. I also feel that Janeway's paradox that we often need adjuvants to raise immune responses is actually very understandable, because when people in the 1960s tried to expose animals, mice in particular, to antigens, they didn't want to take an antigen which was terribly immunogenic, because under whatever circumstances they gave it, they got an immune response. What they selected antigens which were sometimes under some conditions non-immunogenic, and other conditions they were immunogenic. Then they could do things to the non-immunogenic moiety to find out what was required to make it immunogenic, like coupling a carrier to it, or aggregating it. And in this way, they chose antigens which were not very immunogenic. And these actually tended to be homologous vertebrate serum proteins, for which there were not that many helper cells. So they were actually weak antigens from that point of view, and they were chosen to get interesting results. And of course, they needed adjuvants to get an immune response. So I think that PAMPs in danger can certainly influence the size and rapidity of immune responses, obviously, but I suggest they're not generally the critical element as to whether immune responses are initiated. I do think that under some circumstances, PAMP or danger can increase the frequency of cells specific for an antigen, and therefore affect whether you get an immune response or not, even on this model where the threshold number of CD4 cells you need is critical. So how much longer do I have? 10 minutes. Oh, pardon? Yeah, that's fine. So, okay, I won't probably last that long. So, I feel actually that there is a problem in contemporary immunology. I just have a feeling that people are looking at details much too much and not trying to take a broad picture. And one of the things that I'd like to do is I'd like to go back into the really old literature, because I think that there were some experiments done in the old literature which, if you wrote them up now and submitted them to a national funding agency, they'd laugh at you. But I think they're very, very insightful. And actually, there's one particular series of experiments that I never talked about in public before, but I think that it's very, very interesting experiments. They weren't interpreted for many, many years, but they actually tell you from whole animal experiments in the modern context that when you give antigen without adjuvant, you need helpers to induce helpers. So I'm going to go into that. I just think it's... and how this gentleman, Bill Weigel, did these experiments, because he never... When I talked to him, he could never quite explain why he did his experiments, but they were really very, very interesting. So I think this is something we've already talked about a little bit, how you shouldn't confuse immune deviation, immune class regulation with tolerance to self, and that I'll talk about that in my second session. So, second talk. So Weigel did this experiment in 1961, and I'd just like to go through it rather slowly because I think that it's very, very interesting. So first of all, he did it in rabbits with bovine serum albumin, and he challenged them at different times, but it's 3, 6, and 9 months of age. And when you do it at 3 months of age, as you can see in the top line, you get a good response to BSA, and about 10% of the antibody that you get to BSA reacts with HSA. If you take a BSA, if you take a neonatal rabbit, and on the first day of life you give it 100 milligrams of BSA, at 3 months, if you challenge it, you won't get any response to BSA, and of course there won't be any antibody that cross-reacts with HSA. And if you come along with HSA, you find out you get a bit of antibody to BSA and a lot more to HSA, cross-reaction being about 10% again. And we all know what's going on now. It's that the very fact that in group 3, antibody is produced that reacts with BSA means there must be some B cells in that animal specific for BSA. They're not induced by BSA well or at all because there's a deficiency of helper cells specific for BSA. But there are lots of helper cells specific for HSA, so that B cell, which has a receptor that reacts with both BSA and HSA, can be induced by HSA. So that's what we all realize, and that's fine. There is a mistake in line 4 that I've just seen. So if you take a rabbit which has been exposed to BSA, you actually get no antibody that it's correct, sorry. You get antibody that reacts to HSA and quite a bit that reacts again to BSA, showing that in the unresponsive rabbits, you've got B cells specific for BSA and HSA. Pardon? No, when I described group 3, I was describing the result of group 4, and that was my. No. The result is the same, but in group 3, they give no BSA at birth, 1 day after birth. But, all right, okay. It's just telling you that you get the same kind of response. So you've got BSA, anti-BSA, B cells, which also react with HSA, in the unresponsive and in the normal rabbit. I'm sorry, I misspoke when I went through it. And it just happens to be a rather nice experiment that if you give BSA at the same time as HSA, or a bit before, you block the response of the antibody that production that reacts with BSA and HSA, and that's one of the classical experiments suggesting competition at the level of the B cell between activation and inactivation. Now, this experiment was done in 1961, and actually, when I came into the field, this really was pivotal for me, thinking about one lymphocyte, multiple lymphocyte. But Weigel also did an experiment in 1964, and what he did is he took BSA unresponsive rabbits, and at 3 months of age, he gave them 3 shots or 4 shots of HSA without adjuvant, and then he challenged them with BSA subsequently, and they made antibody to BSA. And if he hadn't challenged them with HSA during that intervening time, they would make no antibody to BSA. So somehow, by giving HSA, he'd now made the rabbits able to respond to BSA, and they couldn't without being challenged with HSA. They could respond now to BSA itself, which means that giving HSA 3 or 4 times allowed T helper cells specific for BSA to be expanded up. So then when he came along with BSA, he would get the B cells specific for BSA activated. That's an interpretation, but he did one further experiment which really was a very similar to the counting kind of experiments that Colin talked about. So that's the experiment I talked about. So just to summarize what this must mean, if I can summarize in words what he showed, is that if you take BSA unresponsive rabbits, you give them a series of injections of HSA, that allows them then to respond to BSA in a way that they would not normally be able to respond without that intervening challenge with HSA. If that response that he got of producing antibody to BSA is T helper cell dependent, it tells you that there must be T cells specific for BSA and HSA, which HSA is much better at activating than BSA is at activating. And of course that would make sense if you require CD4 cooperation. And he did an experiment, which he never interpreted, which exactly fits in with that. So this is one from his 1961 paper. So what he did is, we start off with bovine serum albumin. They're unresponsive to BSA. If he challenges those animals with BSA, 9 rabbits, none of them will respond to produce anti-BSA antibody. If he takes an antigen that cross-reacts with bovine serum albumin a lot, then the delivery of BSA, which wiped out the T helper cells almost 100%, but not absolutely 100% at 3 months of age, the sheep serum albumin you'd expect to inhibit most of the, you'd expect the administration of BSA to inhibit most of the T helper cells specific for the normal antigen sheep serum albumin because the two antigens cross-react a lot. And he couldn't break the unresponsive state with those. But if you took pig, which cross-reacts only 32%, 2 out of 5 responded. And if you took human serum albumin, 12 out of 14 responded. And if you think that the reason why you're not making a response on the primary challenge is because you're not inducing helper cells, this really fits in with the evidence, or it fits in with the idea that you require helper cells to activate helper cells. Yes?
What was the route?
The route was all the same for all the different ones. What was it? I'm stumped. I can't remember. But what I do remember is it didn't have any adjuvant. Right, so there's one other, the last thing which from the really older literature that I'd like to talk about is that there's a lot of evidence which tries to address whether you require B cells for the activation of T cells, Th1 or Th2 cells, and really there are inconsistent, lots of inconsistent observations. One well-known one is one of Polly's papers saying that you do not get immune responses in the absence of B cells. No, you do get responses in the absence of B cells. Thank you, Polly. I just want, good. So there is one paper that came out of Janeway's lab, which is very minimally manipulated, which I find incredibly interesting. And that is what he did is he took a strain of mouse in which H2K mice, in which if he immunized it with mouse cytochrome C, he could generate CD4 cells specific for mouse cytochrome C. And I'm sorry. I'm sorry. No. Sorry. I was talking about the next slide, so I apologize. If you're reading that, it won't make sense to you. I went on to the next slide, so I'd like to backtrack and just mention here that both in animal models and human models of autoimmunity, you often see that the initial T cell repertoire is very limited, and then with time it gets broader. And this phenomenon is called epitope spreading, and that's seen in an odd mouse. And what people did, these Kaufman and Tisch, in the early 90s, was that they showed that if you take the first antigen to which you respond to, and at 1 week of age you inject that into the thymus, you stop the generation of T cells specific for that antigen, and you get T cells, you also ablate the T cells for the other antigens, and that phenomenon of epitope spreading and its abrogation by getting rid of the T cell specific for the first antigen are really understandable in the context of CD4 cells, facilitating the activation of CD4 cells. So I just want to bring up one thing possibly for discussion, that with Polly, tadpoles can respond to frog antigens. During metamorphosis, the lymphocytes are hormonally ablated and the immune system regenerates. So that's one way of nature doing it. But we have adolescent antigens, and one of the arguments that Polly, I think, has made, and it's a very good point, is that there may be adolescent antigens which are really novel at a certain stage of adult development. Or if you're really, and therefore you expect to make an immune response against them, or if you don't want to buy into the danger model, you'd have to say they could be expressed early somewhere else by an air-like mechanism. If this is a real problem, I'd like to just bring up whether there's data on pre-adolescents can be immunized against adolescent antigens much more readily than adults, as tadpoles can be against frog antigens. So I imagine Polly will have something to say about that. So Janeway did these experiments where he could induce, by immunizing in complete foreign adjuvant with mouse cytochrome C, T cells specific for mouse cytochrome C, but only under certain circumstances. What he found out, if you gave mouse cytochrome C in CFA, was ineffective. And he then provided evidence that if you gave mouse cytochrome C specific B cells, you could activate those CD4 cells. And what he did is he raised in one mouse with a cross-reactive antigen B cells specific for mouse cytochrome C by using actually human cytochrome C. And then he took cells from the mouse which had been immunized with human cytochrome C, which also had B cells and antibodies specific for mouse cytochrome C, and transferred to a recipient who he then immunized with mouse cytochrome C in complete foreign adjuvant. And what he found out was that if he transferred immune serum, he got no response. Normal B cells or immune T cells, nothing. But if he transferred immune B cells, he got efficient activation of the mouse cytochrome C specific CD4 cells. So that would suggest that I think it's a bit hard to reconcile as one observation. I wouldn't try to base my whole life on one observation, but it's a rather interesting observation, which is certainly explicable in terms of this two-step two-signal model, but might present some difficulties for a danger PAMPA model. So, is there any questions?
Thank you very much, Peter. Questions?
Yes, Polly.
Can I just give just a few comments to? I think no one was sleeping here, but there is some live streaming, so we cannot exclude that someone was sleeping somewhere. So just a few comments. Peter, you Polly contributed a lot to make your model even more popular than it was before, this two-signal model. But just want to point out for people who are not follow completely, is that a major difference is that the signal 2 in the Deter model is given by a dendritic cell, and it's called co-stimulation. Or macrophage, that's true. The macrophage are interesting. But signal 2 is not antigen specific. In the original two-signal model, signal 2 was and is still antigen specific.
Yeah,
So there is a major contradiction there. And this is why Peter told us today about T helper cell giving signal 2. They give signal 2 to B cells, and you would like very much them to give signal 2 to T helper cells as well to keep the system coherent.
So that's the only way I can get non-interference into it, so that responses to foreign antigens will not interfere and help in the activation of CD4 cell-specific for peripheral self-antigens. Yeah.
So the difference so the signal 2 being antigen-specific or not, and actually the nature of the antigen-presenting cell, because to make it possible, the key antigen-presenting cell in your model is a B cell.
Right.
While in Pauli's model, it's the DC or a macrophage. So some major differences there. Okay, now, Pauli.
So let's continue on that vein. Yeah? Because I think B cells are also very important antigen-presenting cells. They're just not professionals. And since I coined the word professional APC, I get to define it. And a professional is a cell that can turn on a truly naive T cell. I think B cells are semi-professionals. They're great at turning on memory T cells, re-stimulating effector T cells, but they can't initiate. And I don't think Peter is saying they initiate either. I mean, at least from that drawing,
I do.
Right?
No. So, Polly, I would disagree with that interpretation of my interpretation.
Okay.
So what I would say is that if you have a dendritic cell or a macrophage, in step 1, it'll cause the cells to divide.
Yeah.
But if there's not a B cell there, they're going to crash, and that's a pathway to apoptosis. So in a real sense, you require B cell to get your first effector cells.
But you don't think, but would you say that the B cell can also do that job that the dendritic cell and macrophages do, which is to do step 1 of the activation?
Because I believe in minimal models, with my introductory comments, I won't, for experimental reasons, I think that's likely right, that the B cell is not efficient in step 1, but for conceptual reasons, I wouldn't go there and make a decision, because I don't say anything unless I'm really forced into it for some conceptual reason.
So it was a conceptual reason that made me first suggest that B cells cannot be professional APCs.
I know.
And it was the same conceptual reason that led Yerna to the network model, which is that if a B cell can be, which I think is wrong, but whatever, if a B cell can be a primary APC, can be a professional APC, can turn on a naive T cell, then you would be turning on all sorts of anti-idiotypic T cells all the time, because there's no way to tolerize for them.
Well, first of all, for that B cell to turn on a naive T cell, according to my model, it has to be activated.
Sure. So it's a B cell against flu, and it presents flu, but it also presents idiotype.
All right, so there are a whole series of experiments done in the 1970s, unfortunately, fairly far back, on the immunogenicity or lack of immunogenicity with anti-idiotypes, done by Iverson and Bretscher.
Yeah, you have to put in a shitload.
No, you have to actually...
And get it picked up at a decent concentration by a macrophage.
No, actually, that wasn't the experiments. The experiments were that if you take a myeloma protein... It was done with myeloma protein, so you can say that that's different from B cells, right? But what they found was that if you take a myeloma protein, you can't make an immune response and add anti-idiotype to it in a complete foreign adjuvant. That's what they found. They found if you TNP'd it and skin painted, you could. And if you give the myeloma protein by itself first, and then you came along, TNP'd the mouse, and challenged with the myeloma protein with TNP on it, you'd block the ability to get anti-idiotype. So they had a situation where hapten carrier was controlling the response to idiotype, and in the absence of the helper, the B-cell idiotype was tolerogenic.
Or, since TNP is an amazingly reactive molecule, that it binds to the idiotype part of the antibody and disrupts it. However, we can do this with almost every experiment. Wait, let's go back a minute. You asked if it can be shown that an organism that is young and not yet expressing an antigen later in life can be immunized to that antigen later that comes up, right? So we used milk proteins.
Which proteins?
Milk.
Yep.
And we immunized prepubescent mice to milk proteins, their own milk proteins. And fair enough, we use CFA, but they are immunizable. Their T cells are immunizable. Their B cells are immunizable, so that you can get antibodies in ELISA that will pick up mouse milk. Actually, it was human milk in a human milk transgenic mouse, knock-in mouse. And you can also get T cells that respond and make cytokines. And yet, when you breed those mice and they start making their own milk proteins, they're fine. And that I don't understand.
I mean, I agree it's an interesting observation, the fact that you have to use complete forensic adjuvant.
Oh, we always use complete forensic adjuvant.
Yeah. I mean, so I think it's open. I wouldn't be dogmatic one way or the other.
Actually, in frogs, you said about frogs. If you prime a frog, if you prime a tadpole to adult frog skin, it doesn't make it through metamorphosis.
I know. I do realize that. I mean, so that's a very interesting thing that Polly just said, because I think that we get ourselves into a tremendous lot of problems in the following sense, and I just would really like to bring it up to see whether you agree. Natural selection arranges for things to work 99.9% of the time with a certain threshold. If you go beyond that threshold, then you're not actually looking at what happens most of the time. And you can get into very non-physiological situations by going over normal thresholds, and they can be very misleading. Do you agree with that?
Yeah, absolutely.
Yeah,
and in fact, I would use that to explain the experiment where putting in MCC-specific B cells allows you to prime T cells that would not normally be primed. And here's what I think is going on there. So Lanzavecchia showed that a B cell specific for an antigen can pick up that antigen at a much...
thousandfold.
Yeah,
low.
Thousandfold better than a dendritic cell, right? So if the tolerance to that antigen, which you, I, Mel, and Colin would probably agree, has to have some affinity cutoff, and that affinity cutoff should have some relationship to how it's presented in the periphery, then my prediction would be that what's happening there is that the B cells picking that up are priming CD4 T cells that were not tolerized and that would not normally be activated.
So what would you say if we use exactly the same level of mouse cytochrome C in complete forensic adjuvant, and we give it to those mice the same way as when we give it with B cells, we get activation. But we don't give the B cells. If we can show that we inactivate those CD4 cells, would you be surprised?
No. Well, yeah, maybe some, but not at the same affinity level.
Well, actually, so these CD4 cells are very interesting because, first of all, they're all IE restricted, and they all recognize the same peptide when you make independent clones of them. So it looks like the mouse has got 99% tolerance to mouse cytochrome C, and there are just a few left over. And the fact that there are a few left over means that if you're interested in counting mechanisms, cell counting mechanisms, they're a perfect example to see what you require.
Except that if you inject a mouse with moth cytochrome C, you also get a fairly, they're not monoclonal, but they're using pretty much V beta 11 or V alpha 11 and V beta 3, I think. So even when you give a mouse a foreign cytochrome C, a really foreign cytochrome C, they still give you a fairly limited T cell response. So the fact that they also do that to mouse cytochrome C isn't maybe
Well,
surprising.
actually, these whole experiments were done by Janeway by, first of all, immunizing with human cytochrome C.
Not mock?
No, human.
Okay.
I've got the paper.
Yeah, whatever.
Human cytochrome C. And what he found out is he got antibody which reacted to mouse cytochrome C, but no T cells at all. And then he took mouse cytochrome C and human cytochrome C and put them together in adjuvant. And then he found out he got mouse cytochrome C specific CD4 cells. And then he took the analysis further and by transfer and showed this. So I mean, to me, I don't think I would never rely on one experiment because we don't understand nature well enough to rely on one experiment. But I do think this is a very interesting experiment.
And it's probably true.
Yeah.
Okay, before the coffee break, I think Bjarne would like to ask a question.
Yes, just a comment about this with B cells as APC compared with, for example, dendritic cells. And I must say I really disagree with Polly. I think there are now several examples. We have published this ourselves with BCR knock-in mice, where certainly the B cells are completely naive and the T cell responders are completely naive. And the B cells are very good APC as long as you ligate the BCR and the B cell takes the antigen in via the BCR. If you give antigen for which the B cells are not specific, they do, of course, not present. And the second thing is that I don't think that Bjarne was very concerned at all with T cells in his idiotypic network theory. That was actually that was the bad thing about this network theory.
Okay, let's please, I think we make a break here because Bjarne will give a talk tomorrow, so we can take this discussion again about B cells, APCs. Thank you again to all speakers, and please come back at 4 o'clock for the next talk by Polly. 4:05.
And then just